With each ovarian cycle, a number of primary follicles begin to grow, but usually only one reaches full maturity and only one oocyte is discharged at ovulation. This oocyte is in the metaphase of the second meiotic division. Spermatozoa are not able to fertilize the oocyte immediately upon arrival in the female genital tract, but must undergo a capacitation and acrosome reaction. So what is capacitation and what happens during acrosome reaction? The two-cell stage is reached approximately 30 hours after fertilization. Once the zygote has reached this stage, it undergoes a series of mitotic divisions, therefore increasing the number of cells. The embryo starts from cleavage until the end of the 8th week. The embryo first undergoes division without growth. Until the 8th cell stage, they form a loosely arranged clump. After the 3rd cleavage, blastomeres maximize their contact with each other, forming a compact ball of cells held together by tight junctions. Compaction segregates the inner cells from the outer cells. About 3 days after fertilization, the compacted embryo divides again to form a morolla. It's a 16-cell stage. The inner cell mass gives rise to tissues of the embryo proper, and the outer cell mass forms the trophoblast, which later contributes to the placenta. About the time the morolla enters the uterine cavity, fluid begins to penetrate through the zona pellucida into the intercellular spaces of the inner cell mass. The intercellular spaces become confluent, and finally a single cavity forms, which is the blastocele. In this slide, this embryo now becomes a blastocyst, and the inner cell mass is now called embryoblast. The outer mass or trophoblast flatten and form the epithelial wall of the blastocyst. Embryonic stem cells are derived from inner cell mass. These cells are pluripotent and can form virtually any cell or tissue type. They have a potential for curing a variety of diseases. The zona pellucida has disappeared, allowing implantation to begin. Trophoblastic cells over the embryoblast pole begin to penetrate between the epithelial cells of the uterine mucosa on about the 6th day. Normally, location is at the anterior or posterior upper 2/3 of uterine wall, mid-sagittal plane. The processes include disappearance of the zona pellucida, exposing invasive trophoblast. Second, embryonic pole is attached to endometrium. Next is the disarrangement and degeneration of epithelial cells and endometrium, and lastly, the sinking of blastocysts into endometrial stroma. At the time of implantation, the mucosa of the uterus is in the secretory phase. By the end of the first week of development, the zygote has passed through the morolla and blastocyst stages and has begun implantation in the uterine mucosa. Implantation is a highly organized process that involves an interaction between a receptive uterus and a competent blastocyst, which can only occur during a limited time span known as the window of implantation. The window of endometrial receptivity is restricted to day 16 to 22 of a 28-day normal menstrual cycle, 5 to 10 days after the LH surge. The uterus then continues into the non-receptive period for the remaining cycle as a late luteal phase until menstruation ensues. Implantation begins with the loss of the zona pellucida, known as hatching, about 1 to 3 days after the morolla enters the uterine cavity in preparation for attachment. Premature implantation could occur if the passage of the fertilized ovum along the fallopian tube is delayed because of tubal damage following infection or premature shedding of the zona pellucida from the fertilized ovum. Ectopic pregnancies occur in 2% of all pregnancies and account for 9% of all pregnancy-related deaths for the mother. The embryo dies about the 2nd month of gestation and may result in severe hemorrhaging in the mother. Anything that hampers or delays the migration of the fertilized ovum to the endometrial cavity can predispose a woman to ectopic gestation. The risk factors would include tubal damage from infection or prior abdominal surgery, history of previous ectopic pregnancies. Smoking may contribute to decreased tubal motility by damage to the ciliated cells in the fallopian tubes, use of hormonal contraception, and 95% occur in the uterine tube. However, most of these are in the ampulla, which is 80%. At the 8th day of development, the blastocyst is partially embedded in the endometrial stroma. The trophoblast in the area over the embryoblast differentiates into cytotrophoblast and syncytiocytotrophoblast. The embryoblast also differentiates into two layers, hypoblast and epiblast. On day 9, the trophoblast shows considerable progress in development, particularly at the embryonic pole where vacuoles appear in syncytium. These vacuoles fuse, forming large lacunae, and this stage is known as lacunar stage. At the embryonic pole, flattened cells, probably from hypoblast, forms a thin membrane, the exosylemic membrane, that lines the inner surface of the cytotrophoblast. These membrane, together with hypoblast, forms the lining of exosylemic cavity or primitive yolk sac. By days 11 and 12, the blastocyst is completely embedded in the endometrial stroma, and the surface epithelium almost entirely covers the original defect in the uterine wall. The blastocysts now produce a slight protrusion into the lumen of the uterus. Lacunar spaces in syncytium form intercommunicating network. The syncytiotrophoblasts cells penetrate deeper into the stroma and erode endothelial lining of maternal capillaries or sinusoids. The syncytial lacunae becomes continuous with the sinusoids, and maternal blood enters the lacunar system and eventually the trophoblastic system, establishing the primitive uteroplacental circulation by the end of the second week. By the 13th day of development, the surface defect has usually healed. Occasionally, however, bleeding occurs at the implantation site as a result of increased blood flow into the lacunar spaces. Because this bleeding occurs near the 20th day of the menstrual cycle, it may be confused with normal menstrual bleeding. The trophoblast is characterized by villous structures. Cytotrophoblasts proliferate and penetrate into the syncytiotrophoblasts, forming cellular columns surrounded by syncytium, and is called primary villi. The secondary or definitive yolk sac is formed as the hypoblast produces additional cells that migrate along the inside of the exosylemic membrane and form a new cavity within the exosylemic cavity. During its formation, large portions of the exosylemic cavity are pinched off, and these can be seen as cysts in the chorionic cavity. The extraembryonic mesoderm lining the inside of the cytotrophoblast is called chorionic plate. The only place where extraembryonic mesoderm traverses the chorionic cavity is in the connecting stalk. With development of blood vessels, the stalk becomes the umbilical cord. The primary villi consist of cytotrophoblastic core covered by syncytial layer. During further development, mesodermal cells penetrate the core of primary villi and grow toward the decidua. Newly formed structure is called secondary villus. By the end of third week, mesodermal cells in the core of the villus begin to differentiate into blood cells and small blood vessels, forming the villus capillary system known as tertiary or definitive placental villus. Capillaries in the tertiary villi make contact with capillaries developing in the mesoderm of the chorionic plate and in the connecting stalk. These vessels establish contact with the intraembryonic circulatory system, connecting the placenta and the embryo. Hence, when the heart begins to beat in the 4th week, the villus system is ready to supply the embryo proper with essential nutrients and oxygen. The difference between the embryonic and embryonic poles of the chorion is also reflected in the structure of the decidua. The functional layer of the endometrium is shed during parturition. The decidua over the chorion frondosum, the decidua basalis, consists of a compact layer of large cells, decidual cells, with abundant amounts of lipids and glycogen. This layer, the decidual plate, is tightly adherent to the chorion. The decidual layer over the abembryonic pole is the decidua capsularis. By the end of the 3rd month, intestinal loops are withdrawn to the body of the embryo. The yolk sac usually shrinks and is gradually obliterated. Extraembryonic space in the cord is obliterated. Allantois Wedelin duct and its vessels are also obliterated, and what remains in the cord are the umbilical vessels surrounded by Wharton jelly. The amniotic fluid is clear, watery fluid, which is 30 ml at 10 weeks, 450 ml at 20 weeks, 800 to 1000 ml at 37 weeks, and at 5th month, the fetus swallows 400 ml. Also, fetal urine is added daily to the amniotic fluid. The amniotic fluid allows the embryo to mold further its body form. It is a shock absorber. It prevents adherence of the embryo to the amnion. It allows fetal movements and maintains temperature. And during birth, it forms a hydrostatic wedge. By the beginning of the 4th month, the placenta has two components: the fetal portion derived from the trophoblast and extraembryonic mesoderm. The maternal portion is derived from the uterine endometrium formed by the decidua basalis. On the fetal side, the placenta is bordered by the chorionic plate. On its maternal side, it is bordered by the decidua basalis, of which the decidual plate is most intimately incorporated into the placenta. The decidua forms a number of decidual septa which projects into intervillous spaces but do not reach the chorionic plate. Because the decidual septa do not reach the chorionic plate, the contact between intervillous spaces in the various cotyledons is maintained. Maternal blood is delivered to the placenta by spiral arteries in the uterus. Erosion of these maternal vessels to release blood into intervillous spaces is accomplished by endovascular invasion by cytotrophoblast cells. Invasion of these cells transforms these vessels from small diameter, high resistance vessels to large diameter, low resistance vessels that can provide increased quantities of maternal blood to intervillous spaces. Cotyledons receive their blood through 80 to 100 spiral arteries that pierce the decidual plate and enter the intervillous spaces at more or less regular intervals. Pressure in these arteries forces the blood deep into the intervillous spaces and bathes the numerous small villi of the villus tree in oxygenated blood. Placental exchange does not take place in all villi, however, only those that have fetal vessels in intimate contact with the covering syncytial membrane. As pressure decreases, blood flows back from the chorionic plate towards the decidua, where it enters the endometrial veins. Hence, the blood from the intervillous legs drains back into the maternal circulation through the endometrial veins. The full-term placenta is discoid in shape, 15 to 25 centimeters in diameter, 3 centimeters thick, and 500 to 600 grams. This is torn from the uterine wall 30 minutes after delivery, then expelled. The functions of the placenta includes formation of a barrier between mother and embryo, delivery of nutrients and oxygen. It removes waste from embryonic blood, and it also becomes an endocrine organ which produces hormones and takes over the corpus luteum. So it produces estrogen, progesterone, HCG on the first 2 months, and somatotropins. Monozygotic twins develop from a single fertilized ovum. They result from splitting of the zygote at various stages of development. The earliest separation is believed to occur at the 2-cell stage. Splitting of the zygote at the early blastocyst stage, the inner cell mass splits into 2 separate groups of cells within the same blastocyst cavity. The 2 embryos have a common placenta and a common chorionic cavity but separate amniotic cavities. The separation occurs at the bilaminar germ disc stage just before the appearance of the primitive streak, which is rare. This results in formation of 2 partners with a single placenta and a common chorionic and amniotic sac. Although the twins have a common placenta, blood supply is usually well balanced. So read further on this in a reference book. Dizygotic twins, or the fraternal twins, they are from 2 ova fertilized by 2 different sperms, which results in 2 embryos, 2 separate placentas, 2 separate chorion and amnion, but sometimes placentas are fused. This is a picture of the placenta of twins. This is a picture of the placenta of triplets. Approximately 75% of conjoined twins are female, and 70% are fused at the thorax, thoracopagus, or abdomen, omphalopagus. The union can be in the frontal, transverse, or sagittal plane. The 2 main categories of conjoined twinnings are symmetrical and asymmetrical. In symmetrical or equal conjoined twins, there are two well-developed babies. But in asymmetrical or unequal conjoined twins, there is a small part of the body that is duplicated, or an incomplete twin is attached to a fully developed twin. An especially rare condition occurs when one incompletely formed parasitic twin is dependent on the well-formed one. This is known as heteropagous twinning. One twin is not fully formed, the parasitic twin, and it is dependent on the well-formed one. A malformed head, upper limb, and large ears are present in the parasitic twin. Pygopagous is known as twins that join at the buttocks, ischiopagous join at the ischium, craniopagous join at the head. Bicephalous or dicephalous means that two heads are present on a single trunk. In the condition parapagous or diprosopous, twins have lateral union of their trunks so that both faces are forward-looking in the same plane. Pygopagous is a term used when the twins face in opposite directions. The sacra are fused and the twins may share a portion of the spinal cord. In addition, the rectum and perineal structures are usually fused.